Good day, I'm Paolo, and here we will be talking about tissue engineering with our group. Okay, let's give a little introduction. Regeneration means the growth of a damaged or missing part of the remaining tissue. As a result, humans can regenerate some organs such as liver if the part of the liver is being lost by a disease or an injury. If the liver will go back to its original size, though it's not always an original state for our skin, be renewed and be repaired. Unfortunately, many other human tissue don't regenerate, and a goal in regenerative medicine is to find ways to kick-start tissue regeneration in the body or in engineering replacement tissue. And some parts of our body can repair themselves quite well as after injury, but others do not tend to repair their bodies for it suffers a heavy injury. It can regrow when lost during an accident or birth defect. But some animals can regrow or regenerate whole body parts, and that's where tissue engineering starts to kick in.
So this is the background or the history of tissue engineering. In 1665, Hooke discovers a small hole in the cross-section called cells, and his book called Micrographia. In 1838 to 1839, Sladen and Swan formulated the so-called cell theory based on microscopic findings. In 1858, Virchow described his ideas about cell formation with the now famous words omnis cellula e cellula, saying to raise from already exciting cells, proving Sladen and Swan of spontaneous cell generation. And then Virchow represented these ideas about regeneration, stating that tissue regeneration is dependent on cell proliferation. And this led for the researchers to focus attention towards the more fundamental cellular level. Thursch attempted to grow skin cells into granulation, or the small particles which are in the wounds. In doing this, he discovered the important influence of granulation tissues on wound healing in 1874. And then Loeb first reported the idea of growing cells outside the human body in 1897. From that point on, many researchers experimented with growing cells in vitro. And then Harrison in 1870 to 1959 was the first to grow frog ectodermal cells in vitro in 1907, thus developing the first neuronal tissue culture line. This was followed in 1912 by Carrel, who was able to grow pieces of chick embryo in various media, which he initially maintained for 85 days, and subsequently for years. Later, interest aroused in growing cells instead of complete tissues. In 1916, Rose and Jones discovered that trypsin is capable of degrading matrix proteins, thus separating cells. And trends changed toward expansion of cell types throughout the 1940s and soon after. Then much research was performed, which led to ability to grow tissue-specific cells in vitro. Enders contributed greatly to the use of human embryonic cells in 1952. In 1998, the development of stem cells formed the basis of modern tissue engineering. Research since 1998 has focused on identifying the differential capacities of embryonic and adult stem cells and how to influence and accompany this differentiating pathway. Aside from this, research focuses on the problems associated with the expansion and autologous and allogenic differentiated cells, as a large number of cells are needed to generate a relatively small volume of tissue.
So, after knowing the history and introduction of tissue engineering, these are the importance of tissue engineering. As we all know, there are many importance of tissue engineering, but these are the most important objects of tissue engineering. The first one, fewer livers available for transplant than there are patients waiting for new livers. Second one, a strategy for construction of the organ must be developed. The third one, tissue engineering holds the promise of producing better organs for transplant. And next one, possible to correct many otherwise incurable genetic defects. And lastly, artificial tissues can revolutionize healthcare.
So here are the applications for the tissue engineering. First, there is a replacement of lost skin. Second, there is a replacement or repair of defective or damaged bones, connective tissues, or intervertebral discs. Third, there is a replacement of worn and poorly functioning tissues. Also, there is a replacement of damaged blood vessels. And restoration of cells that produce critical enzymes, hormones, and other metabolites is also an application. And for the benefits, the first one is there is a reduced cost, reduce of less expensive treatments. Also, there will be dramatic improvements in treatment outcomes. And lastly, there will be an improved quality of life for patients.
So the advancements of tissue engineering. Unique micro-robotic technique to assemble the components of complex materials. Complex materials is the foundation of tissue engineering and 3D printing. This approach uses untethered magnetic micro-robotic coding for precise construction of individual cell-encapsulating hydrogels, such as cell blocks. The micro-robot, which is remotely controlled by magnetic fields, can move one hydrogel at a time to build structures. This is critical in tissue engineering, as human tissue architecture is complex with different types of cells at various levels and locations. When building these structures, the location of the cells is significant in that it will impact how the structure will ultimately function. Compared with earlier techniques, this technology enables true control over bottom-up tissue engineering. This will revolutionize three-dimensional precise assembly of complex and heterogeneous tissue engineering building blocks and serve to improve complexity and understanding of tissue engineering systems.